Now let's look at the mechanism of DNA replication at the molecular level. Here we have again that diagram. Now let's look at the molecular view of DNA replication. I know I keep showing you this slide over and over again. This has important information we can look at from different perspectives. We have talked about DNA being anti-parallel, two ends of DNA being different. One is the 5' end, the other is 3' end, and the prime end basically mean the carbon atom, which is 3' carbon atom, which has the hydroxyl group. Here I will again point that out. Here is the 3' end because the carbon atom number has a 3' hydroxyl, which is available to make a bond with the next nucleotide. This is the 5' hydroxyl. This is the 5' end. Here we don't have a hydroxyl group. The 5' carbon is attached to a phosphate group. So this is basically, again, we are looking at these pentose sugar molecules, deoxyribose molecules. Here's the molecule of deoxyribose. Number 2 carbon is with the hydrogen atom. It does not have hydroxyl. RNA pentoses or the ribose sugar has a hydroxyl group on carbon atom number 2. So again, carbon atom number 1 is the place where the base is attached. I have inverted it so that it is easier for you to understand. So this is the 5' end. This is the 3' end. Now that we have reviewed our DNA molecule, let's look at how DNA molecule grows. As we have already established, the two ends of DNA molecule are different. There is a 5' end and the 3' end. New addition of new nucleotides on a DNA polymer only happens at the 3' end. This is the 3' end. There is no known enzyme that can add a nucleotide at the 5' end of the DNA chain. So this strand here, this is serving as the complementary strand or the parental strand. The new nucleotides are added which are complementary. Here, for example, we have C and G. The next nucleotide here is also a C. So the correct nucleotide which will come in front of this G is a C. So the molecule that puts new nucleotides on a growing DNA chain, the DNA polymerase, it will read the complementarity, it will read the DNA molecule, the parental DNA molecule, it will take the correct nucleotide, it will attach the 5' end of this nucleotide to the hydroxyl group which is present here. So this is the reaction. This carbon hydroxyl basically attacks the phosphate group, and this bond is formed, new bond is formed. Please notice the two phosphate groups are released. This molecule is called the pyrophosphate. Remember, the phosphate group is a negatively charged entity. So you basically have a spring, a compressed spring. The potential energy is in the compression of the spring. The two negatively charged entities are very close together. So when this bond breaks, it releases huge amount of energy, which is used to make this bond the phosphodiester bond of the growing DNA chain. So this is basically the molecular mechanism of DNA replication. The next point is, does the machinery which is replicating DNA, is it moving along the DNA, or is the machinery that is replicating DNA stationary and DNA is basically threaded through this machinery? We have two ways we can accomplish the same feat. Chromosomes have specific sequence of DNA which serves as origin of replication. It's a special sequence of nucleotides which is recognized by machinery that replicates DNA. It binds there and it can proceed with the process of DNA replication. Now, what is moving and what is stationary? Initially, people used to think that it is the DNA replicating machinery which is moving along the DNA molecule. Now people think it is the other way around. The machinery is actually large, complex. It is stationary and DNA is threaded through this machinery. Now, I would also like to point out that bacterial chromosome only has one origin of replication. It is a circular chromosome. In humans, we have linear chromosomes, and each chromosome has several origins of replication. So a chromosome is threaded through several DNA several molecules, several complexes that replicate DNA. Additionally, I would like to point out that we need two complexes of DNA. We are making two strands of DNA. In one case, the replication machinery will be moving in this direction, all things being relative. Either the DNA is being threaded through this direction. Actually, it will be easier to understand this concept if we say that DNA machinery is moving, DNA polymerizing machinery is moving. So one machinery is moving in this direction along the DNA molecule, other machinery is moving in the opposite direction, as you can see on the slide. So again, this linear chromosome has more than one origins of replication. Bacterial circular chromosome has only one replication of origin, and human chromosomes have several of these. Next, we will continue our discussion of DNA molecule.